Our final lecture of Unit 1 is Neuroplasticity and Aging. The central nervous system has an amazing ability to change. Change can be a result of the environment and experience or due to injury or illness. Some of the changes that take place include a change in function, something that we often call functional reorganization, or the change in the amount or type of neurotransmitter that is produced and released, or a physical change in structure and connections between neurons. One form of neuroplasticity is to alter the strength of the synapse. How strong is the signal between neurons? We can either enhance or potentiate that signal, or we can depress that signal. Either of these can be short-term effects or long-term effects. They are known as sensitization, short-term depression, otherwise known as habituation, long-term potentiation, or long-term depression, also known as long-term habituation. Sensitization often occurs due to repeated application of a stimulus that is typically of high intensity. It is short-term, can last up to a week or so, and then you return to the previous level of response. The effect of sensitization is an enhanced response in the postsynaptic cell to a particular stimulus or a class of stimuli. Sensitization is what occurs when you cram. So when you cram for an exam, you are actually increasing the response of the postsynaptic neurons to the particular stimulus, the content of the course, for instance. But again, it is temporary and it is reversible, which means it might help you get through the current exam, but it's not going to help you at the end of the semester for the final. Nor is it going to help you next semester when you have to retrieve the information that you are expected to have learned, or in the clinic when you need to retrieve that information in order to assess your client and to come up with appropriate OT interventions. So you want to try and minimize and avoid cramming as much as possible. With the sensitization, again, this is going to give us a potentiation or an enhancement of synaptic transmission. It frequently occurs with presynaptic axon terminal changes, such as in having an increased number of synaptic vesicles containing a greater amount of neurotransmitter. So your presynaptic neuron produces and transports more neurotransmitter to the axon terminal. We create additional synaptic vesicles for that neurotransmitter so that when the action potential travels down the axon and depolarizes the axon terminal, a greater amount of neurotransmitter is released into the synaptic cleft. The greater the amount of neurotransmitter released into the synaptic cleft, the longer it's going to take to clear out that neurotransmitter from the synaptic cleft. So the longer period of time that neurotransmitter can be activating the postsynaptic neuron. Short-term depression or habituation is typically due to prolonged exposure to a benign stimulus. This would be repeated exposure to a low-intensity stimulus. That's what I mean by benign. So we have a low-intensity stimulus that is repetitive and with a high frequency. So there's a very short period of time from one stimulus to the next. So action potentials are basically being formed in the presynaptic neuron at a relatively rapid rate. With short-term depression or habituation, we see it can come on very quickly, but we also have a spontaneous recovery of the sensitivity of the synapse with rest. You can also see depression or habituation when there's lack of activation of a synapse or when we have exposure to an extremely strong stimulus. I've put it here with short term, but most commonly those two types of stimuli responses would be seen or would produce a longer term effect. So really those are probably better to go under long-term depression and habituation. And I would have you actually take lack of activation and exposure to a very strong stimulus. And I would have you cross them out here and put them on your slide for long-term depression. Because the short term seems to be the primary response really when we have a repeated exposure to a high frequency, low intensity stimulus. An example might be street noise, cars going by while you're trying to study. The cars are going by on the road, you can hear the cars, but you kind of tune them out rather quickly. That would be an example of a short-term depression or habituation. The effect of short-term depression is an attenuation or a reduction of synaptic transmission. We don't know a lot about how it works, but some of the mechanisms that have been proposed include a decreased release of neurotransmitter from the presynaptic neuron. This may be the result of a reduction in intracellular calcium, so that there is less calcium in the axon terminal to initiate exocytosis, or a depletion of neurotransmitter because we have high frequency stimuli. Because we have a high frequency of action potentials, the soma cannot keep up with the production and transport of neurotransmitter down to the axon terminal. So we're essentially using up our neurotransmitter faster than we can make it. It's also been proposed that there are postsynaptic changes in the sensitivity of the receptors on the postsynaptic membrane, and that they just don't respond to the neurotransmitter binding as well. Long-term potentiation is what happens when we have learning taking place. Long-term potentiation is what you want to encourage when you are taking your coursework. It involves two phases, an early phase and a late phase. The early phase is really an enhanced sensitization. In addition to having greater neurotransmitter released from the presynaptic neuron, we also here will see insertion of receptors into the postsynaptic membrane. Now this process has been best delineated in glutamatergic synapses. This would be synapses between neurons where the presynaptic neuron releases glutamate as its neurotransmitter. Glutamate binds to receptors that result in excitatory responses in the next cell. And glutamate is interesting because it requires a couple of different types of receptors in order to have its effect. It requires NMDA receptors, and NMDA is short for N-methyl D-aspartate, and AMPA receptors. And AMPA has the long, convoluted name of, let me think here, alpha amino 3 hydroxy 5 methyl 4 iso xalopropionic acid. I want you to know AMPA and NMDA. I will have questions written out where you will not have to write out in a short answer what AMPA and NMDA stand for, unless that information is already given to you. Okay. Now, on an open book where you can look up what these are and how to spell them, I would still expect you then to know them. But on a closed book assignment or an exam, don't worry about having to know exactly what NMDA and AMPA stand for. But do know that NMDA receptors and AMPA receptors are glutamate receptors. We have some synapses that have NMDA receptors in the membrane, but either no AMPA receptors or few AMPA receptors. And again, for that glutamate to really have an effect on the next cell, we need both. So these are called silent synapses. When you have the higher intensity stimuli activating the glutamatergic neuron, releasing glutamate. That glutamate binds to the NMDA receptor. If we're at a silent synapse, the response to the NMDA receptor binding with the neurotransmitter is an influx of calcium into the postsynaptic cell. As the intracellular calcium levels increase, it causes AMPA receptors that are in the cytoplasm to be inserted into the postsynaptic membrane. So this postsynaptic cell that at its silent synapse, it has AMPA receptors ready and waiting, but they're not yet in the membrane. They're in vesicles within the cytoplasm. The calcium triggers the cell to insert those AMPA receptors into the membrane. Now that we have both NMDA and AMPA receptors in the membrane, it allows for the opening of additional ion channels that will create EPSPs with sufficient summation to generate action potentials in the next cell. So we've now sensitized this existing synapse and activated it. In the later phase, we actually see the synthesis of new proteins, the synthesis of additional receptors, and the creation of new synapses, something called synaptogenesis. Synaptogenesis means birth of synapses. So we are making new additional synapses. It all starts with that influx of calcium, and this needs repetition. We have to have the influx of calcium not just a couple of times, that'll create that early phase, but continued, repeated influxes of calcium. These repeated influxes of calcium and insertion of AMPA receptors into the membrane, leading to additional activity in the cell, triggers these various biochemical processes that cause the production of additional receptors and activates genes. Calcium is very important in a variety of processes in cells, including gene activation. And so we now start turning on genes that are responsible for producing additional dendrites and additional dendritic spines. And dendritic spines are like these little outcroppings on dendrites that are the perfect location for a synapse. The process of making additional dendrites and adding dendritic spines is called dendritic arborization because it looks like a tree that's growing additional branches. With synaptogenesis, we are looking at a lifelong process. So long-term potentiation doesn't stop at any certain age, but it continues across the lifespan. It is strongest when we're young, but it continues all the way through advanced age. It has been shown to be enhanced by physical activity and physical exercise. So staying active is important. It's also important to remain active while you're trying to learn new things. So as you are studying, you should take frequent breaks and move and create some physical activity. And in order to get the repeated stimuli, the repeated influx of calcium, you need lots of repetition. So you're actually much better off to study for short periods of time over a number of days as opposed to studying a long period of time in just a single day. That's more of that cramming kind of a thing. But if you really want new synapses to form and long-term retention of material, you need to study shorter periods every single day. And that is really the ideal way of learning. There's an interesting study that was done using sea slugs that helped to illustrate that synaptogenesis is required and the production of protein in order for that is needed for synaptogenesis is required. So they took sea slugs, these are rather large creatures, and they put them in these cages that had an electrified grid on the bottom. in a particular area of that cage. And the sea slugs learn to avoid this electrified grid because, they go over the grid and they get a shock. And they would actually learn to go around it and avoid it as they were moving around in the cage. But if these sea slugs were given some kind of a treatment that prevented the synthesis of these new proteins required to create new dendrites, then they did not learn to avoid that electrified grid, and they would continually cross over the grid and get shocked. Long-term depression also is something that takes place lifetime. Long-term depression is often seen following the decrease in firing of a synapse over a long period of time, or a repeated exposure to very high frequency action potentials, and extremely high intensity where they are likely to be harmful. Long-term depression is also known as long-term habituation. It involves the removal of receptors from the postsynaptic membrane. So remember, short-term depression or habituation was really involving making a current synapse less effective by reducing the sensitivity of the receptor or by decreasing the amount of neurotransmitter available at the synapse. But with long-term depression, we actually remove receptors from the postsynaptic membrane. In addition, genes that are required for the production of proteins that are needed to help maintain synapse health are turned off. So proteins have a limited lifespan. So as the proteins degrade, we don't have the proteins that are needed to help maintain the synapses. And we start to see degradation of the synapses themselves. So short-term effects, basically, whether it's potentiation or depression, involve enhancing synapses that are already present. Long-term effects involve either creating new synapses or eliminating synapses that were present. When you actually fully eliminate a synapse, we call that process pruning. Pruning is also used to describe the removal of dendritic spines, the removal of dendrites, or actually removing entire neurons. So when you have the death and removal of neurons or any parts of a neuron, that is known as pruning. Neurogenesis would be the birth of new neurons. We only really see this on a functional level in a couple of areas of the brain, and that would be in the hippocampus, an area of the brain we'll talk about quite a bit in the final unit, but it's very important for making new declarative memories, or memories of things that you can verbalize, things that you can talk about easily. Those would all be what we would call declarative memories. So essentially, understanding your neuroscience is declarative memory. And then another area is the olfactory nerve itself. And these are, we do see immature neurons in these areas that are then able to undergo mitosis and creation of new neurons. But mature neurons cannot undergo mitosis. And so in most areas of the nervous system, we do not have these immature neurons. We have mature neurons, and so we do not see new neurons being made. So if you lose neurons, you just have that many fewer neurons available. We do have stem cells, and it seems that when there is injury to neurons, the stem cells do start to divide and to mature into new neurons. However, there are several processes that occur along with the injury that seem to prevent these stem cells from moving forward and actually maturing into neurons. Oligodendrocytes give off particular chemicals. There are some other chemicals. So there seem to be certain things that really retard the ability of these stem cells to move forward. Research is trying to find how to get the stem cells to survive at the site of injury so that they can create new neurons. But at this point, it's still early. So for now, we're going to say that we cannot undergo neurogenesis with the exception of those two areas I mentioned. The other thing to keep in mind is that only neurons that make a synapse and an active synapse survive. So this will be important when we talk about regeneration of neurons as well. Neurogenesis in the hippocampus and olfactory nerve has been shown to be retarded by certain medications like those involved with chemotherapy, and as well as by stress. So people who have chronic stress will have more difficulty creating memories for learning. So they'll have a little more trouble with that. And individuals that are undergoing chemotherapy often complain of something they call chemo brain or chemo fog, which is the medication is interfering with their memory capacity. Neurogenesis, though, in the hippocampus and olfactory nerve are enhanced by physical activity and exercise and an enriched environment. Another good reason to be active. So let's talk about what happens if we have injury to a neuron. If the area of the neuron that is injured is the soma, unfortunately, that means the death of the cell. When the cell body itself is injured, the cell body and its parts, the axons and dendrites, swell up and burst open. This causes the release of calcium and stored neurotransmitter from that neuron. These contents are toxic to the adjacent cells. So this starts causing the adjacent cells to swell and burst open. And so we get this kind of cascading effect. And so oftentimes you'll have a central area of injury that then spreads to the adjacent areas. The reason that it stops is that your brain, in its smartness, brings in glial cells that help to kind of wall off, mostly we're talking microglia and astrocytes, that help to wall off the injured area, preventing those toxic contents from just spreading further and further and further and further. If you have ischemia to the area of the cell, this results, if it's a glutamatergic neuron, can lead to the release of large amounts of glutamate. And too much glutamate actually causes death of the postsynaptic neuron. So it may not be specific, I've damaged this cell by crushing it. It could be that it's lack of blood supply that causes the damage. And then it puts out this huge amount of glutamate. So too much of a good thing is not a good thing. As I said before, neurogenesis is limited to only a couple areas of the brain. So if you lose a neuron, it will not be replaced when it's anywhere in the nervous system. If the injury is in the axon, it's really going to depend if we're talking peripheral nervous system or central nervous system. If we're talking the central nervous system, then we're likely to have regeneration unless the injury is limited to just a few axon terminals. In the case of just a few axon terminals, you can probably regenerate those few terminals that are lost. But if you damage the axon itself, then this neuron is going to die. And that seems to be due to a number of factors, including the release of neurite outgrowth inhibitor or NOGO from oligodendrocytes, some other chemicals that retard regeneration, and also the lack of certain neurotrophic factors that are needed to support neuron health. nerve growth factor is one. So the lack of things that are needed for neuron health, plus the presence of chemicals that actually impair neurons and their recovery, really keeps neurons in the central nervous system from being able to regenerate. But if we're in the peripheral nervous system, now it's a different story. So we're going to talk about injury and recovery when the injury is to the axon in the peripheral nervous system. The first thing that happens is you get degeneration of the distal segment, which is called Wallerian or orthograde degeneration. So I have an injury, everything distal to that begins to degenerate. That's the Wallerian or orthograde degeneration. The proximal segment, the part that is attached to the soma, degenerates a little bit. Usually, though, it's only for 1 to 2 nodes of Ranvier from the point of injury. This is called retrograde degeneration. If the injury to the axon is greater than about 15% of its length from the soma, then that cell is likely to survive. So if I had a cell that was 100 centimeters from the cell body to its axon terminal, and it was damaged 10 centimeters from the soma, it's probably still going to die even if it's in the peripheral nervous system. But if it's 20 centimeters from the cell body, it's likely to survive because it's greater than 15% of its length away from the soma. And most likely it has to do with this retrograde degeneration and how much of that is the damage products go back to the soma and start causing damage to the soma itself. As I mentioned, if we're in the central nervous system, the cell is probably going to die. There is some research that says this is not necessarily the case, but right now we're not seeing any relevant regeneration without us artificially going in and producing or providing some exogenous methods of treatment. And this includes adding nerve growth factor into the central nervous system where it doesn't exist. So as I mentioned, astrocytes and oligodendroglial cells or oligodendrocytes produce NOGO, which is neurite outgrowth inhibitor. There are other chemicals, some we call suicide enzymes, that contribute to the cell death. You get scarring by astrocytes and microglia. These were intended to help wall off the injured area so we don't get that progressive death of cells, but can also then form glial scars that prevent the injured neurons from regenerating. And then failure to have presence of factors that we need to help enhance and maintain cell health, such as nerve growth factor, all contribute to the death of CNS cells. The process of recovery in a peripheral nervous system neuron, however, that is at least 15% of its length away from the cell body, involves the formation first of what's called a growth cone. This is a little axonal sprout. It can occur up to hours after the initial injury, and it elongates very slowly, approximately 1 millimeter a day or about 1 inch a month. One thing that it can help you to in the limbs is you can kind of trace where that person's at based on the return of certain sensations or motor functions. You can kind of tell how that nerve is recovering. It will eventually then reconnect to its target and form a synapse. If it doesn't form a synapse, the cell is going to die, because remember, we have to have a good active synapse for the cell to survive. Schwann cells do not produce NOGO like the oligodendrocytes do. In fact, they produce trophic factors such as nerve growth factor that are needed for the health of that growing and recovering neuron. They also form tunnels that basically guide the axon to its target. Now, sometimes the tunnel doesn't go to the original target and things can go awry a bit, and that can make the neuron not functional, even if it manages to create a synapse. But in some cases, and in many cases, we will successfully get back to the target. Some of the reasons for failure, though, of the regeneration in the peripheral nervous system might be a sensory neuron finding a tunnel meant for a motor neuron, or the other way around. The longer the distance from where the injury occurred to the target, the more likely something's going to interfere with its ability to regenerate that full length. Scars in the area, connective tissue scars that literally block and physically block the neuron, the axon, can prevent regeneration. And again, the location of the injury along the axon. Where is it on the axon? The closer to the axon terminals, the more likely regeneration is to take place. If you were to have injury to a nerve under really, really good conditions, we would expect about 80% of those neurons within that nerve to successfully regenerate in an ideal circumstance. Here is a pictorial of axon regeneration in the peripheral nervous system. So you can go ahead, you have a blank slide, so you can do your own drawing. So you have your original neuron, it's synapsing on its target cell, and there's an injury. And that's where I put the X as the location of the injury. Because of the injury, we see Wallerian degeneration distal to the injury. So everything distal to that neuron is beginning to degenerate and break down. And then a short retrograde degeneration for probably 1 to 2 nodes of Ranvier or so occurs on the proximal segment. A growth cone then forms, so this axonal sprout, and then it eventually grows following Schwann cell tunnel back to the target cell, where it forms a synapse, and we now have full regeneration of that neuron. So now I've got a brainstorm for you. I want you to think about this, and I want you to go ahead and throw it up on the discussion board. See what your thoughts are and what your classmates' thoughts are. Would a central nervous system neuron whose axon is injured at its midpoint, so we're 50% of its length from the cell body, be expected to regenerate? Why or why not? So now I want to talk about some more specific mechanisms of neuroplasticity. Altered input, whether it be an increase in input or a decrease in input, can result in functional reorganization. Interruption in a pathway is more likely to result in unmasking of silent synapses, in collateral sprouting, or in regenerative sprouting. Functional reorganization is when the functions of neurons change. They may change as far as the part of the body that they respond to, or they may actually change based on the type of stimulus that what they used to respond to, they might now change to responding to a different type of stimulus. We see this most markedly in the cortex area, but we can also see it subcortically, such as in the thalamus. Our representation of our body and our environment can be modified by repetitive sensory input. So the more you use something, the larger the area of the brain that is dedicated to it. It can be changed by learning and experience. It can also be changed by injury. So if you decrease use of a body part, for instance, then the area of the brain that is dedicated to it will be reduced. So again, cortical representation for a repetitive task, whether it be sensory input or motor output is going to increase. But if you have a lower frequency of either sensory input or motor output for an area, then that representation is going to decrease. There have been some reassignment of neuron functions that has been noted. For example, the occipital lobe and the back of the brain is where we have our visual regions. People who are born blind or who become blind, we actually see the neurons that have been dedicated for vision actually begin to respond to other sensory stimuli, such as sound or touch. Touch is probably a big one, where individuals who learn Braille, we will see activity of the neurons in the occipital lobe that on a individual with normal vision will only respond to visual input and not the touch of Braille. So it's amazing what the brain is capable of doing. For functional reorganization, we have to be talking about intact areas of the brain. Neurons cannot change their function or start responding to things they didn't before if the neurons are no longer, if they're not surviving. If those neurons have died, they obviously cannot take on a new function. So when you're thinking functional reorganization, be thinking of this is taking place in intact areas of the central nervous system. So here's just an example of someone who had an injury that resulted in amputation of the index finger. It could even be an injury that involved immobilization of the index finger, because these changes can start occurring very quickly. So you have someone who has maybe their index finger completely immobilized, or you have someone who had an amputation of that index finger. Originally, there's a part of your brain where there is a group of neurons that respond to sensation and movement of your thumb. Let me pick one or the other because they're actually different parts of the brain. So let's talk sensation. So you have a part of your brain that responds to sensation on your thumb. You have another part of your brain that responds to sensation on your index finger, and another part of your brain that responds to sensation on your middle finger. Now these areas are right next to each other on the brain, but if I touch my index finger, the thumb neurons don't really do anything, and neither do the middle finger neurons. But after that index finger has been immobilized, so now I touch the cast, but I can't feel it on the finger, or it's been amputated, so now there's no finger to touch, the neurons that used to respond to touch on my index finger now start responding instead to touch on my thumb or to touch of my middle finger. So the area that's represented by the thumb becomes much larger, as does the area representing the middle finger. Now, if it were the case of the immobilized finger, if I were then, after sufficient healing time, allowed to begin moving that finger again, and I could, now the cast was off and I could touch it, and so there's starting to be sensory input again, we would see the brain reorganized back to what it started with, where the index finger will have an area of cells of neurons specifically dedicated to the index finger. The area for the thumb will be smaller again, as will the area for the middle finger. So it can change in either direction. This is often a good thing, but sometimes it is not such a good thing, and it can lead to problems such as phantom limb pain or chronic low back pain can be enhanced by some functional reorganization in the brain. Silent synapses we've already talked about in relation to long-term potentiation. We have these synapses that basically are there because there's these NMDA receptors, there's enough activity that they don't get pruned away. And we can call upon them though when we need them. So we might need them because we're learning something new and we need to create a synaptic network between neurons in order to get that information to be in our brains long term. Or we might need them because the original area that information was transmitted has been damaged and that information cannot go that route any longer. It's like a detour. We need an alternative route because the bridge is permanently out on the road that we used to take. Silent synapses can also be masked synapses where they're either overpowered or inhibited by other neuron pathways. So sometimes you've got a very strong pathway and it's also putting out inhibitory synapses to another potential pathway. And so that second pathway is weak. It doesn't reach summation to threshold frequently because there's a lot of inhibition there. So these stronger, typically more direct connections are what you generally follow in order to transmit the information that you need to transmit. So there's a couple different reasons for that you might have a silent synapse. But these silent synapses can be unmasked when that stronger, more direct connection is disrupted. So here's an example of silent synapse. The idea of I've got a couple different ways that I can transmit information. The top two neurons here we'll consider to be a more direct route to where we're going. And it's going to typically be strong. And there might be that in the neuron where I've got the silent sign is that either there are no active AMPA receptors in that membrane, or it's being inhibited by other neurons so that the information goes the quick, direct, strong route. But now that pathway, that quick, direct, strong pathway has been disrupted and we've had a neuron that's died. We now insert AMPA receptors into the membrane, or we eliminate or reduce the inhibition onto that second neuron so that we now have a good functional synapse and we have an alternative, although less direct route, we have an alternative route in which to take so that we can still get the information to where we need it to go. So again, the best way to think about it is a detour. Collateral sprouting is another mechanism of neuroplasticity. And this is where you have a neuron that dies and neighboring neurons then put out sprouts to the surviving neurons. We have seen evidence of this both with spinal cord injury and with polio. So what we're looking at is we have death of neurons causing denervation of a postsynaptic neuron. It's lost its presynaptic input. So a neighboring neuron forms a collateral to go over and create a synapse on that postsynaptic neuron that was denervated. So that postsynaptic neuron now gets information and is able to transmit the information as it had previously. I mean, ideally and obviously these neurons are having the same function. So if it's a motor neuron to a motor neuron, not a motor neuron causing a collateral onto a sensory neuron, that would not make sense. Okay, and they're going to a similar location, a similar target, maybe different muscle cells within the same muscle, for instance. But this is an example of collateral sprouting. A presynaptic neuron is damaged, denervating the postsynaptic neuron. A neighboring neuron puts out a new collateral to that postsynaptic neuron. This is different than regenerative sprouting. With regenerative sprouting, you have damage to a postsynaptic neuron and the presynaptic cell, but typically we're just talking about, again, maybe it's axon terminals. We're not talking about way up into the axon. Might just have lost a few axon terminals along with the postsynaptic neurons. That surviving presynaptic neuron then creates a new synapse with an adjacent uninjured neuron. This is going to occur much less often in the central nervous system because of all the problems we have with regeneration. And again, only if the loss involves only a few axon terminals. This is, though, an area that's highly exciting in research as they are working with putting in nerve growth factor and other types of things to encourage regenerative sprouting. And they're having some nice successes. We can see a couple of processes that occur on a temporary basis following denervation of a neuron. These are denervation hypersensitivity and synaptic hypereffectiveness. Denervation hypersensitivity is a temporary upregulation of postsynaptic receptors. So when you increase the number of receptors in a membrane, that's called upregulation. When you remove receptors from a membrane, that's called downregulation. So when we have long-term potentiation, we start off by upregulating the number of receptors in the membranes for the early phase, and then eventually we create additional synapses. With long-term depression, we are downregulating the receptors in the membrane. The more receptors we have, the more neurotransmitter molecules we can grab hold on, the more ion channels we can open, the more the response. And the reverse, of course, is true for downregulation. So when a neuron is denervated, so you've got a postsynaptic neuron that's lost the presynaptic innervating neuron, it will temporarily upregulate the number of receptors, hoping to grab onto some neurotransmitter molecules from other uninjured neurons that are also synapsing on it. Synaptic hypereffectiveness is a temporary increase in the amount of neurotransmitter that is released at a particular synapse. This occurs when you have a small number of axon terminals damaged in a presynaptic neuron. Your soma continues to produce the same amount of neurotransmitter. But now there's fewer axon terminals for it to be transported to. And so the remaining axon terminals have a greater amount of neurotransmitter than they used to have. Let's say we had 5 axon terminals and we were producing 25 molecules of neurotransmitter. So each axon terminal received 5 molecules of neurotransmitter to release. But we've now damaged 2 of those axon terminals. We now still have 25 neurotransmitter molecules being produced, but there's only 3 axon terminals. So each axon terminal now is going to have, what is 25 divided by 3? 8 neurotransmitter molecules, and then one's going to get a 9th one. So we are able to release more neurotransmitter because the soma is continuing initially to produce the same amount of neurotransmitter, even though there are fewer remaining intact axon terminals. Your book talks about some of what we have learned regarding rehabilitation and its effect on neuroplasticity. Early rehab is very important for limiting functional loss and for limiting loss in adjacent undamaged regions. It's also important to enhance functional reorganization, to allow undamaged areas to modify their function, to take over some of the function that was lost by the injured areas. However, if your rehab is too intense and too soon, this actually has a detrimental effect on neuroplasticity. The other thing I think that's really important is to understand that chronic injury does not mean neuroplastic changes cannot take place. When I went to school and got my PT degree, We were told, whatever you've got at a year, that's it. You're not going to get any further changes. Nothing can happen after a year. So don't even bother. We now know that's not true. Um, there are many times that we see people that are quite far out from the initial injury and make small improvements. It's true that in the initial phase following the injury, that's when the greatest and fastest improvements are going to take place due to these neuroplastic changes. But that does not mean that down the road we will not see any type of neuroplasticity taking place. So to increase your learning and the learning of your patients, these are some of the things that you need to make sure to do. You need to make sure to get adequate sleep. There are many, many sleep studies that show that even a couple of hours reduction of sleep at night retards learning and retards long-term potentiation. Your brain needs the recharge. Keep stress to a minimum, so exercising, yoga, taking time out to be with your family, watching a movie, whatever it might be, to try and keep some balance while you're undergoing this. I know it's a stressful time in your life. You've got, the coursework, you've got the work that you're doing in the clinic, you've family. I know you're being pulled in lots of directions. Believe me, I just finished my PhD. Took me 6 years working full time. I know what it's like. I got my master's degree when my children were little. I know what it's like to be trying to work, have a family, going to school. It is a stressful time. So you need to make sure that you're putting in some balance into your life. Exercising. Physical activity, as I've mentioned a couple of times now, it enhances long-term potentiation and enhances learning. Even if it's just a matter of getting up for 5-10 minutes and just taking a quick walk around the house, anything that gets you moving is a good thing. It also breaks up your studying so that you're not overstudying for too long of a period of time and essentially getting more of the cramming kinds of response as opposed to the long-term potentiation response. Frequency is a key here. So if the stimuli comes too quickly together, we start to actually see depression occurring as opposed to potentiation. So if you break up your studying into small groups, you will find that you're now getting the repetition, but the frequency between the studying of the same material is increased. And so you're much more likely to potentiate instead of attenuate. Frequent repetition. We want to get that calcium coming in on a repetitive basis. So again, you want to study this every day, not big chunks just in one day, but yet being specific with your task. So you're not just all over the board, but you're repeatedly studying the same stuff over and over and over again. It's not just I look at this page, then I'm not going to look at that page of my notes again for another 2 weeks. Making sure you're eating well, and again, taking frequent breaks is important. We do know that for learning, you also need to be able to attend. You have to attend to whatever it is that you're looking at or working on. If your attention is elsewhere, you're not going to learn. And you require certain neurotransmitters such as norepinephrine for arousal and attention. So you're not falling asleep on your book. You need to be aroused and you need to be paying attention. And that takes norepinephrine. And if you're too focused on your studying for too long a period of time, you deplete the norepinephrine. And remember, neurotransmitter depletion is one of the methods behind depression, short-term depression or habituation. So again, you need to have frequent breaks, and you need to know this both for yourself, but you also want to keep these things in mind when you're working with your clients in the clinic. And now very quickly, we're going to talk about aging. If you haven't been taking breaks already while listening to this lecture, I want you to stop now and make sure you take a break. Get up and move. Maybe even when you come back, you're going to study something else. Do your kinesiology and then come back to your neuro. As we get older, our brain shrinks. We have a small loss of neurons. It's estimated at about 0.2% every year. These are preferentially large diameter neurons, which are our fastest neurons, and we'll talk about that next unit. We are going to lose some of our voluntary motor neurons. These are called alpha motor neurons that are going to go to our muscles. And that's partly because these are large neurons. These are some of our larger neurons. So this results in atrophy and decreased strength in muscles. When you look at an older individual, you can see that their limbs are, not as robust as they were when they were younger. We have an increased size of the motor unit. A motor unit is a single alpha motor neuron or motor neuron and all of the individual muscle fibers it innervates. And what happens is that we start getting larger motor units. We lose some neurons. Some of those remaining neurons are going to adopt some of the muscle fibers that were denervated. And so that motor unit now just got bigger. I used to innervate 10 muscle fibers, but now I'm innervating 13 because I adopted 3 of them that lost their nerve. And as a result, I now have a bigger motor unit. This means that we see less fine, precise motor control. So you see less fine abilities in older individuals than in younger. We see a greater loss of cross-sectional area in our high threshold muscle fibers. These are fast twitch fibers. These are the ones that respond explosively. And so older adults are more likely to have difficulty responding to perturbations quickly. So you bump into them, it's harder for them to respond quickly and right themselves because of the loss of high threshold motor units. Or to have if you ask them to do something as fast as possible, they have a harder time being able to generate the high speeds than as a younger person. This process of losing these motor neurons is slowed down, although it's not stopped with exercise. So again, activity, it helps both cognitively, it helps the way our nervous system works all over, it helps us with our motor neurons. So the entire nervous system really benefits from physical activity. Unfortunately, we can't stop the aging process. We can slow it down. We can't stop it. We also see things like the myelin sheath on our nerves gets thinner in some areas, and this is somewhat controversial depending on the nerve, but we definitely see more defects in our myelin, and we see retraction of the internodes. Remember, the internodes are actually those areas of myelin. So they're the areas between the nodes of Ranvier. So it's the area of axon that is covered with myelin. We see retraction of these internodes. So the myelin gets thinner and it kind of pulls in on itself. That means that the nodes of Ranvier become wider. But the areas on the nodes of Ranvier that are now exposed don't have high concentration of ion channels in them. So there's these areas of axon with few ion channels, and they're no longer insulated. This results in slower action potential transmission speed. We end up with a reduction in nerve conduction velocity because our saltatory conduction is impaired by having these exposed areas of axon that do not have high concentrations of ion channels. Remyelination is also slowed down. We might need remyelination because of injury to the myelin, but also remember that myelin is made up of oligodendrocytes and Schwann cells, and all cells have a limited lifespan and have to be replaced. So when you lose those cells and you need new ones to remyelinate that area, that whole process is slowed down. There's also a reduction in the amount of type 1 collagen in the coverings of the neurons. Type 1 collagen is our strongest type of collagen, and collagen is a protein that's involved in connective tissues. So it gives a nice strong covering to our nerves. And if we have a reduction in the strong type of collagen and it's replaced by a weaker kind of collagen, then that nerve is more likely to sustain an injury. We see a small decrease in the number of synapses when we get to old age, somewhere around 10 to 30% fewer synapses than what we had. So remember, when we're born, we have lots of neurons and relatively few synapses, considering what we're going to get. As we mature, we lose a bunch of neurons, but we have these very complex networks that now form with lots of synapses. And as we get old, we lose a few of those synapses, so we don't have quite as many as we did. We have decreased sensitivity of sensory receptors. We see that most people, as they get older, end up getting glasses. They might need hearing aids. So we see some of our sensory receptors are less sensitive than they were before. The brain puts out less melatonin. This is a substance that's required for sleep. So sleep tends to be shallower and shorter. And we have a reduced but not absence ability, remember, for long-term potentiation and synaptogenesis. So while its learning ability is decreased, it certainly is not eliminated. And by doing brain exercises and cognitive exercises and lifelong learning, you will find that you can really help to minimize this reduction in long-term potentiation. And now just to talk about a couple of pathologies. Dementia is the loss of cognitive and intellectual function that we often associate with aging, although it's not specific to just aging. It is many times progressive. Symptoms include impaired memory, judgment, and intellect. It has many causes. Everyone always thinks of Alzheimer's disease, but that is just one cause of dementia. Dementia is a result of AIDS. It could be vascular in nature. It could be due to alcoholism. It could be due to deficiency in vitamin B12. There are many things that can result in dementia. And then specific to Alzheimer's disease, this is a very sad disease. It's associated with a massive loss of neurons. Remember, normal loss with aging is 0.2% per year. This is 2.8% per year, more than 10 times as much. Tends to begin in the temporal lobe, and the temporal lobe, we will learn, is an area involved with recognition of people and things, and then as well as with the formation of memory. So we see the memory problems, the difficulty in recognizing other people, in recognizing things, and then it spreads to all areas of the brain. Ventricles tend to get very large. Essentially, they're just filling up the space where the neurons are dying.